We'll wrap up chapter 8. We'll get started with chapter 9 and see how far we get. So, we left off on Thursday talking about different kinds of horizontal gene transfer. And we talked about 2 of the 3 major ones. So, we talked about transformation, how microorganisms can sometimes just pick up DNA from their external environment and incorporate it into the genome. And the organisms that can do that, we say, are competent. So an organism that is capable of picking up DNA from the external environment is competent. The other mechanism we talked about was conjugation, and that's where microorganisms will make a structure called a pilus. So one microorganism will have the ability to make the pilus. The other microorganism will receive the pilus, and a plasmid can be exchanged that way. And not really even exchange, a plasmid can be given from one cell to the other. It is not reciprocal, there's not a trade. Okay. So the final mechanism by which bacteria can get DNA from other bacteria in their own generation is called transduction. Then there are 2 major types of transduction. There's generalized transduction and specialized transduction. And what transduction means moving DNA from one cell to another through the use of a bacteriophage. A bacteriophage is a virus that infects DNA, or bacteria. A virus that infects bacteria and can move DNA. So, when we talk more about viruses, maybe starting next week we'll get into viruses. When we talk more about viruses, we'll talk about the idea of viruses being very, very simple, often consisting of just genetic material and proteins. Bacterial viruses, or bacteriophage or phage, are very simple viruses. Have you guys done the bacteriophage lab? Ooh, okay, so you get to play with them in the lab. And don't worry, they can't infect you. They can only infect bacteria. What they do is they recognize a host cell. So for them, the host is bacteria. And they inject their DNA into the host cell. That's the infection. And again, when we talk more about viruses specifically, we'll talk about how bacteriophage are different from viruses that typically infect animals, and how those cycles are very, very similar, but also in some ways different. So a bacteriophage inserts its DNA into the host cell, and that starts the infection process. What happens is, just like any viral infection, that DNA basically starts to take over the host cell's machinery. So instead of the bacteria being able to replicate its chromosome, it replicates the phage DNA. It replicates the viral DNA. Instead of the bacterium being able to do transcription and translation of its own genes, it does transcription and translation of viral genes. So it makes viral proteins instead of bacterial proteins. And when this happens, this leads to the breakdown. Some of the genes that viruses can make allow them to break down the bacterial chromosome. So they can break down the bacterial chromosome while making their own proteins. And what happens is, in the cell, the phage are assembled, is what we call it. The phage DNA is assembled inside of the phage protein. And eventually what you have now is this bacterial cell that has basically just become a phage factory. All it makes is phage. But again, right, you're talking about a limited space, and the phage is making as many copies of itself as possible, so you get the cell to explode, which releases all of the phage to go on and infect new cells. What can happen that leads to transduction is that during the process, we're looking at step 3 here, during the process where DNA is being packaged into the phage, sometimes host DNA gets packaged instead of viral DNA. And they're trying to illustrate that where these phage have purple DNA inside of them, which represents the bacteria, rather than pink, which represents the virus. When that virus goes on to try and infect a new cell, it injects bacterial DNA, which doesn't cause an infection. Instead, the cell can incorporate that new piece of DNA into its chromosome, and now it has acquired a new piece of DNA through horizontal gene transfer. And you know, this idea of viruses being able to incorporate their DNA into host chromosomes is something that we do see a lot with animal viruses too. This is a really important evolutionary mechanism for viruses, to be able to incorporate their DNA into a host chromosome. This is just kind of a viral accident. It's a dead end for the virus because it is not moving its own DNA, it's moving bacterial DNA. That doesn't help the virus. These cells are not going to make new virus. But that's kind of how this mechanism evolves, that it is important for many viruses to be able to integrate their DNA into a host chromosome. Well, you just integrated the wrong DNA. So I told you there are 2 different types of transduction. The first is generalized and the second is specialized. So what's the difference? Well, the difference is what piece of bacterial DNA gets packaged. The one that I just showed you, it was just a random piece of the bacterial chromosome that accidentally got packaged inside of a phage. That's generalized. It doesn't matter what piece of bacterial DNA went in there. It was just a piece of bacterial DNA. It was just random. It could have been anything from any part of the chromosome. So again, we're looking at that life cycle of the phage. The phage will bind to the donor cell and inject its DNA. It takes over the host cell machinery, which eventually leads to the host chromosome falling apart. As the phage are being assembled inside the host cell, sometimes accidentally, host cell DNA gets packaged instead of viral DNA. Again, this might seem like an evolutionary dead end, but we're talking hundreds or thousands of phage that can burst out. If a few of them are wonky, you can still cause a really great infection. So let's say these are not desired, right? Because it's not going to allow the phage to continue its cycle. This phage is a dead end because it only has bacterial DNA. So that phage carrying the bacterial DNA infects the new host cell, and that new cell, in theory, could incorporate that DNA into its chromosome. So again, the reason that we call this particular process generalized transduction is that any piece of the bacterial chromosome has an equal chance of being packaged. It doesn't matter what part of the chromosome it is, everybody's got a chance. So it could be any gene. And then they introduce another term on here that we'll also talk about as we start talking about chapter 9. And that term is a recombinant cell. So a recombinant cell is going to be a cell that contains DNA from 2 different individuals. So at the bottom, the new cell that's been infected has its own lavender chromosome, but it also has a piece of this darker purple chromosome. So now it is a recombinant cell. That dark purple came from a different individual. The lavender was its own. So now you have a cell that has DNA from 2 different individuals, a recombinant cell. Okay, let's see here.
In transduction, DNA from the chromosome of one cell is transferred to another cell via a replicating virus. A virus that infects bacteria is called a bacteriophage or phage. There are two types of transduction: generalized transduction and specialized transduction. Generalized transduction, as shown here, is initiated during the lytic cycle of virulent bacteriophage. The donor DNA can be any fragment of the bacterial chromosome. Specialized transduction, which is shown on a separate animation, is initiated during the lysogenic cycle of a temperate bacteriophage, and the donor DNA is a very specific part of the bacterial chromosome adjacent to the prophage. At the beginning of generalized transduction in bacteria, a bacteriophage attaches to a bacterial cell wall and inserts its DNA. The phage commandeers the bacterial cellular machinery to synthesize the new phage DNA and produce phage proteins. In order to completely convert the bacterium into an efficient virus-producing factory, some of the phage proteins break the bacterial DNA up into small pieces. Normally, phage DNA is coated with phage protein to form new virus particles, but sometimes pieces of bacterial DNA are mistakenly packaged in phage proteins. These sloppily packaged phage particles carry bacterial DNA rather than phage DNA and are called transducing phages. When a transducing phage, composed of phage proteins and bacterial DNA, infects a new host, the phage injects the bacterial DNA into the new bacterial host. The infected bacterium sometimes incorporates this DNA into its own chromosome through recombination, resulting in the transfer of genes from the donor bacterium to the recipient bacterium.
Okay, so what questions do we have about generalized transduction? Okay. Yes.
In general, do you know what the rate of, the virus mistakenly packaging bacterial DNA is?
Yeah, that's a great question. I actually don't know what the rate is. I imagine it might depend on the virus, and it might depend on the size of the genome of the bacterium. As far as we know, basically, and I actually had this discussion today in bioinformatics. One of the grad students has never taken microbiology, so when we talked about the idea that, viruses can infect bacteria, he was like, wait. He's like, wait, bacteria can get an infection? And I'm like, oh yeah. So there's a lot we don't know about it, but as far as we know, everything can be infected by a virus. But some organisms have really small genomes, like less than 500,000 base pairs. I'm sorry, yeah, less than 500,000, so like 500,000 or so. Some bacteria have very large genomes, 10 million or more. I imagine probably the larger the genome, the more likely it would be that a random piece could be picked up. But I'm actually not sure if anybody's ever studied the rate of that. But the phage do tend to be pretty specific. Just like, oftentimes we talk about infections that infect only people or only mammals. Bacterial viruses can also be pretty specific for that. So yeah, it's a really good question, and I'm not sure. I know with some human infections, the rate of insertion into the chromosome is actually really high because that is the actual strategy for the virus. Like HIV, it's 100%. They all get into the chromosome because that's the strategy for replication. In terms of this, yeah, I don't know. But when you're talking on the order again of the way that you'll measure this and the way you'll see it in lab is when you infect you'll make a lawn of bacteria, and when you infect them with the phage, what you see is clearing. So you'll see these little zones of clearance, and we call them plaques, and that's where one cell was infected, and then the infection just spread to all the neighboring cells. So overall, they're pretty efficient because we do see that spread, but yeah, I imagine there's probably a good number of them that are messed up. Other questions about generalized transduction? Okay, so specialized transduction was alluded to in the previous video. So specialized transduction is a little bit different, and it takes advantage of an infection strategy that only some phage have. So not every phage is capable of doing this, but some of them are. And some of them can enter what is called a lysogenic cycle. And I'm not supposed to write with these. Let's try that one to see if it works. Whatever. Okay, so they enter what's called a lysogenic cycle. So what happens during the lysogenic cycle is it's a little bit different from what happens during the cycle we showed before. They call that the lytic cycle. The lytic cycle is very simple. Virus gets in, virus replicates, virus explodes the cell and moves on. Yeah. Pretty simple strategy. Lysogenic is a little bit different. So yeah, lysogenic, virus gets in. And we'll see this in the animation. Virus gets in, but instead of replicating itself, bursting out and spreading, the strategy here is purposeful integration into the host chromosome. So it doesn't get in, take over everything, and explode the cell. It gets in, gets into the chromosome and sits there. And it sits there. And it sits there. But while this is happening, the host cell is replicating, right? So you have one cell that got infected. The virus inserted its genome into the host cell. The host cell replicates, and every time it replicates a copy of that viral genome. So you start with one, and overnight you have trillions of cells that all have a copy of this piece of viral DNA in their chromosome. So integration into the host chromosome, replication of the host. Then something happens, and the trigger can be different for every type of cell. Often it's a stressor. Something is happening to the population that's causing a little bit of stress. And what will happen is that the viral DNA will pop out. So the viral DNA pops out, and then you get explosions, right? Once the viral DNA comes back out of the chromosome, it will take over the host cell, lead to its own replication, and then kill the host cell. But now, instead of one cell releasing hundreds or thousands of phage, you have millions or billions or trillions of cells that have all just... So the lysogenic cycle allows them, it's just a different type of replication strategy. They can lay low, allow the population to grow, and then eventually explode out. So how does this have anything to do with transduction? Well, what this has to do with transduction is that sometimes, and they say rare, I don't know the frequency when they say rare. Again, when I talk on the scale of trillions, it may not be that rare. But rare event is that when the viral DNA is popping out of the host chromosome, it accidentally takes a piece of the host chromosome with it. So it's not a mispackaging event. It's not swapping out viral DNA for host DNA inside the phage. It's putting in a viral DNA that has a little bit of host with it. It's not a clean break when it excises out of the chromosome. It grabbed a little host chromosome with it. And when that happens, all the phage that are released now are a little combo of phage and host. And then when that phage goes on to infect a new cell and it integrates into the chromosome, it integrates that little piece of DNA from the previous host. So we call it specialized transduction because it's not any piece of host DNA that gets packaged, it's the special piece of host DNA that was right next to where the phage DNA was. And so depending on where the phage had integrated into the host chromosome, which again can be random or can be very specific, there could be, here they're showing you galactose metabolism genes, or an antibiotic resistance cassette, or what we were talking about in bioinformatics, a pathogenicity island, a whole region of DNA that contains a lot of different virulence factors. So it's specialized because it takes a very specific piece of DNA, the one that was right next to the phage.
Specialized transduction is initiated during the lysogenic cycle of temperate bacteriophages. Recall that during the lysogenic cycle, phage DNA becomes incorporated into the bacterial chromosome. The phage DNA is now called a prophage and can be transferred to subsequent generations of cells each time the DNA is replicated. During induction, when the prophage is excised from the host chromosome, it occasionally takes with it some of the bacterial chromosomal genes. This phage DNA, with the attached bacterial genes, then begins the lytic cycle, where it's replicated and becomes packaged into new bacteriophages. The bacteriophages are released when the cell bursts. One of the bacteriophages attaches to another bacterial cell, the recipient. The phage releases the DNA into the recipient cell, and it becomes incorporated into the recipient's chromosome. Now the recipient cell has a prophage plus new bacterial genes from the donor cell. This recipient cell is a recombinant cell because it's received DNA from another bacterium. Specialized transduction can be used to transfer antibiotic resistance from one bacterium to another. The donor bacterial cell has a resistance gene we will call R. As a prophage is excised from the donor cell, it takes the R gene with it. Copies of this DNA are made and packaged into phage particles. When one of these phages infects a recipient cell that is not resistant, the R gene is transferred to the recipient's chromosome. Now this cell becomes antibiotic resistant due to acquisition and expression of the R gene.
And so we know this happens because we can see it in the genome. We can see the evidence of this having happened. And I mean, we see it in bacterial genomes, we see it in our own genomes. When you analyze the genome of an organism, you can look at the evolutionary history of viral infection, where sometimes the prophage just stays incorporated and never really becomes reactivated. And over time, it loses its ability to become reactivated, but it still had provided this benefit to the recipient cell. And we even see that there are human diseases that are caused by bacteria that have undergone a process like this, where the toxin that the bacteria produces is actually produced by the virus DNA that it now holds. So diphtheria, which is a respiratory infection, is mediated by diphtheria toxin, which has a history as being from a prophage. Cholera, which causes really massive amounts of diarrhea, like liters a day, is mediated by a toxin that is from a prophage. So again, we see this in medically relevant bacteria, we see this in environmental bacteria, and by looking at the genome and analyzing, well, where did these pieces of DNA actually come from, we can get really cool insights into evolutionary history. And again, we have a lot of evidence of this in our junk DNA. We have those human endogenous retroviruses. So retroviruses are things like HIV that their whole strategy is to integrate into the host chromosome. We have huge portions of our DNA that are actually viral infections throughout our evolutionary history. So this type of strategy Even though, again, it sounds very random, and it's like, how would this really work? It has worked, and it continues to work on really large evolutionary scales. So it's really cool phenomenon. Okay, questions?
Yeah, for like that example in the video that talked about antibiotic resistance coming from this process, like if it's coming from like this virus that's affecting it and has a small piece of nice DNA, but the rest of it's going to kill it eventually. You mentioned there's also the possibility it inactivates and then stays dormant completely. Is it more often that it's like that, where nothing happens that's bad, or does it come with some good parts and then also eventually dies?
I think it really depends. It depends on how long I would say it probably depends on how long the bacteria holds onto the prophage before the prophage tries to reactivate, because the longer the cell holds onto it, the more likely it is that it acquires mutations that prevent it from being able to do the excision of itself. And again, we look for in these types of regions of bacteria, what we look for are things like integrase genes that allow it to integrate into the chromosome. We look for transposase genes that would allow it to pop in and out. So sometimes they just, over time, accumulate enough mutations. And with bacteria, we're not talking about huge timescales here, but it accumulates just enough mutations that it no longer has the ability to pop in and out. Yeah. I imagine there's a lot of failed opportunities of this too, where it still does pop out and the cell loses, the cell loses because it dies. Yeah. Other questions about specialized transduction? Okay, cool. So we also can talk really briefly, like 3 slides, about things called mobile genetic elements. In bacteria, we classically mean things like plasmids, but we also could talk about things like transposons, and we have transposons as well. One of my favorite female scientists is Barbara McClintock, and she was the one who discovered this phenomenon. She called them jumping genes. She's found them in corn. So this is a really, again, not unique to just bacterial idea, that there are pieces of DNA that are capable of moving. So we call them mobile genetic elements. Usually when we're talking about something like a plasmid is going to be able to move from one cell to another, especially through something like conjugation. But even, transformation, they could pick up a plasmid that way. With transposons are usually elements that are in the chromosome. They're like a region of the chromosome. They can sometimes be moved from one cell to another, but oftentimes, like I said, they called them jumping genes. They can move from one chromosome to another. They can move within the same chromosome. Sometimes they can copy themselves and then insert that new copy somewhere else. So they have these really cool abilities that can cause new phenotypes. But of course, classically in microbiology, we talk about plasmids. We talk about plasmids because this is where we see a lot of antibiotic resistance that can spread from one cell to another, again, especially through conjugation. But broadly, a plasmid is an extra chromosomal piece of DNA, and it is self-replicating. It has to be. If a plasmid doesn't have its own origin of replication, it will not be replicated because, right, primase doesn't know where to go, DNA polymerase doesn't know where to go. It has to have an origin of replication. They're most often circular, but again, there are a lot of exceptions. There are organisms that have linear plasmids too. So yes, they're really useful for bacteria to spread, especially antibiotic resistance. But as we move into chapter 9 on biotechnology, we really commonly use plasmids in biotechnology and genetic engineering. So in a natural setting, a plasmid that can be spread through conjugation does have to have, right, that F+, that fertility factor, the genes that allow it to make the conjugation pilus so that it can spread from one cell to the next. Some plasmids can have genes for really unique catabolism. And this is where it gets useful for things like bioengineering or genetic engineering. You could put like hydrocarbon metabolism on there and then use the organism for bioremediation of things like oil, like cleaning up pollution. On this one, they're showing a hypothetical gene for mercury resistance, like it allows it to tolerate mercury. And then plasmids, even in the genetic engineering in the lab, right, will have one or more antibiotic resistance cassettes. We use them in the lab, and we'll talk more about this in chapter 9, for being able to select for cells that have the plasmid, because we look for the antibiotic resistance. But again, they can share antibiotic resistance that way. And so this is like the bonus plasmid. It has like everything. So it has the genes necessary to make the pilus, to do conjugation. It has mercury resistance. It has antibiotic resistance to sulfonamides, streptomycin, chloramphenicol, and tetracycline. So it's a multi-drug resistant plasmid. And those are all very different mechanisms of action. Typically in nature, when we see antibiotic resistance on plasmids, we don't see so many genes on a plasmid, but certainly in a laboratory setting and you're using it for genetic engineering, you could have multi resistance on a single plasmid, because we can use that for selection. And then, of course, the last mobile genetic element are going to be the transposons. These are the pieces of DNA that can move. And again, sometimes like the piece can cut itself out of the chromosome and go somewhere else. Sometimes it can make a copy of itself and that copy can go integrate somewhere else, but they move. Usually a transposon will have what are called recognition sequences, these little repeats right here. That allows them to recognize DNA, integrate, like put themselves in there. They'll often have genes that allow them to cut themselves out and then glue themselves back in. They can be, this plasmid or this transposon right here is engineered to have what's called a kanamycin resistance cassette. We use this a lot in bioengineering to do what's called transposon mutagenesis. How do we know what a gene does? Right? Sometimes we don't. Everything we know about what genes do is by studying those genes. And the best way to study those genes is to figure out, well, what happens when the gene is gone? So how do we know? In humans, we can't, right? We don't do that. We don't engineer humans. But in humans, we can look at their DNA and say, oh man, all of these people have this same disorder and they all have an issue with the same gene. This gene probably plays a role in causing this disorder. In bacteria, right, we can play with them. We can take away their genes. And so you can do what's called transposon mutagenesis. You take a bunch of bacteria, you give them a transposon, you see where it inserts. And then you see what different phenotypes happen when you interrupt a gene. That's how we learn about what genes do. So it's just kind of I don't know what's going to happen. Let's make thousands of mutants and see what happens. So it's a really common technique in microbiology and genetic engineering. So one of the challenges, though, is sometimes where these things integrate is really bad. Maybe for bacteria, whatever, it integrates into a gene that they need for survival, so they die. And maybe we don't care that much because they're just bacteria. But what happens if, in humans, they integrate into a gene that we need to stop the cell cycle when something has gone wrong? Or a gene that we have that's critical for our metabolism, then our cells die. Or if it goes into, a gene that stops the cell cycle, then that's the development of cancer. So we tend to not use transposons when we do human gene therapies. And we'll talk more about human gene therapies in chapter 9, because we need it to be more specific. And there have been a lot of examples of doing genetic engineering in people to fix genetic conditions that have gone very, very badly. So this kind of non-specific integration, like kind of wherever it integrates is where it integrates, can be really, really challenging to deal with. So we typically don't use transposons for human medicine. Questions about anything in chapter 8 before we start a little bit of chapter 9? Okay, cool. Let me find my slides. Okay, so chapter 9 is on biotechnology and DNA technology. Biotechnology is a really incredible field for biologists to get into. There are a lot of job opportunities in these fields, not just in human medicine, but in agricultural biotechnology, food, right, microbiology. So any of you who are considering fields outside of like human healthcare, there are a lot of opportunities for biotechnology, and the biotechnology in this region is really growing. So again, there's a lot of opportunity, especially in the food and beverage industry, but you know, bioremediation of pollutants, all sorts of stuff. But of course, if you're interested in like human biotechnology, there are lots of options there too. So these are some of the things that we'll cover over the next couple of days, but let's just jump into it. What do I mean when I say biotechnology? Well, biotechnology is just the use of a living system to do something, generally to make some sort of product. I'm using a living organism to make a product. It could be food, antibiotics, vitamins, enzymes or proteins for people who need them, vaccines. So the use of a living system to make a product. Oftentimes, when we're doing biotechnology, what we have done is create a recombinant organism. We have put DNA from one organism into another, and we have asked that cell to do something for us. So what do I mean? Well, you may know that many diabetics are insulin-dependent diabetics. Okay, insulin for diabetics used to be isolated from cows because they're big. Do you know how expensive it is to isolate and purify insulin from cows? Very, very expensive. How do we get enough insulin for all the people that need it and have it be as affordable as possible? Not counting the, US healthcare system. We make it in bacteria. So the human insulin gene has been cloned into bacteria, and bacteria then make human insulin. They can do it at a much larger scale than cows. It can be purified much more readily. So in theory, when someone like Mark Cuban is like, insulin shouldn't be more than like $4 or $5 a dose, he's right. It should be that cheap because it's that easy to make. But that's what we mean by doing recombinant DNA technology, for example, for medicine. How many of you have been vaccinated against Hep B? Healthcare field, if you haven't, you will be. They use yeast to make that vaccine because they put viral genes into the yeast. The yeast express viral proteins that can then be used to make the vaccine. So we have lots of reasons that we would make recombinant organisms. I mean, when I say like, oh, I'm gonna put a piece of DNA from this species into this species, it kind of sounds like a mad scientist, right? That's not what we're doing here. These are really, unique genes that provide a really important downstream effect, and we make them at a larger scale. But again, people sometimes get fussy and they're like, oh, GMOs. I'm like, we've been modifying organisms genetically for our entire history. Like, you know what a wild banana, like an actual banana, it's like mostly seed. Seedless water, I mean, how does a seedless watermelon reproduce? It can't, right? Wild, like original, like corn is not good. That goes over better at Fresno State because they grow their own corn and they're very like, ooh, our corn is so good. I'm like, well, it wouldn't be if it hadn't been genetically modified. I mean, how many of you have a dog that looks something like one of these? I mean, I have a couple that look like that. There's no reason a dog should be this short but this long. I mean, besides that they're so freaking cute. You know, they were bred to hunt badgers. Like, so humans have been genetically modifying organisms for food, for, you know, hunting. Cats. You want to bring a bobcat home? Probably not. I mean, they're cute, but rip your face off. Your cat might think about ripping your face off, but he's not going to. Right? Through domestication, through selective breeding. Plants and animals look very different now than they did. So these are genetically modified. Now, are they recombinant? Not necessarily, because we didn't go in and physically mess with the DNA. Instead, we selected traits we wanted and bred and bred and bred and bred until we got the result we wanted, which again is a dog that's like this tall but this long. Okay. Or this one that doesn't have any hair. Those Chinese crested, man. That one, yeesh. No offense, but they always win the ugliest dog contest. So if you have one of those, it's not just me, okay? So what do you do then if you find an organism that has a trait you want, and you want to play with it, let's say? So you can do what's called selection. You can look for a microbe that occurs naturally and has a specific trait that you're looking for. For example, we know that there are microbes that are capable of degrading oil. How do we clean up oil spills? We use bacteria. So we know that there are bacteria that have evolved the ability to break down those types of hydrocarbons. We know that bacteria are able to pull soluble uranium out of water and turn it into insoluble uranium in areas that are contaminated with like nuclear waste. So you have to find them. You select for organisms that have these abilities. You can cause mutations. You can induce random mutations, maybe through transposon mutagenesis, or here they give an example of using like any kind of mutagen and force them to evolve the trait because you change their DNA. But a lot of this is random or luck, right? You found a bacteria that's capable of degrading oil. I mean, if you know where to look, you can find them. You've induced a random mutation that gave you a beneficial phenotype. It's luck. It's chance. So what do we do when I don't want to actually have to like mutate, like zap a bacterium until it makes insulin. It's never going to happen. That's never going to happen. Well, we directly then modify the organism. And there are a number of different ways that we can do that. So we create specific changes within the organism. One of those tools that we will talk about is CRISPR. CRISPR's kind of the big one. That's the one that everybody's like, ooh, holy grail of how we're going to do genome editing. Okay. But what did we do before we had the holy grail? Well, we made recombinant DNA. Okay, and we're going to go through this figure kind of like panel by panel. But, you know, at the end of the day, by taking a gene from one organism and putting it into another, we can start to do things like make plants that are resistant to being eaten by herbivorous insects. So there are plants that make a toxin that kills things like caterpillars and other herbivorous insects that would destroy the crop. But the toxin is not toxic to humans. We can take enzymes that we know help bacteria degrade hydrocarbons and put them into bacteria that work better, that grow faster. We can add enzymes that allow better clothing preparation for fabrics, like different colors, different fabric processing. And of course, we can put products in bacteria that we can then use for people. I used an example of insulin for people who have insulin-dependent diabetes. And for people who are deficient in human growth hormone, where do they get human growth hormone? Not from humans, from bacteria. So this idea of finding a gene that we're interested in and putting it into the system that will allow us to get as much as possible is really the goal here. So broadly, what we do when we're doing genetic engineering is we start with a plasmid, right? This extra chromosomal, self-replicating piece of DNA. And there are literally thousands of plasmids that you can work with in a lab. And they all have different purposes. Some of them allow you to swap one gene for another. Some of them allow you to express levels of bacterial proteins, like high levels. Some of them allow you to express high levels of eukaryotic proteins. Some of them, might have enzymes that allow you to degrade hydrocarbons. All of these plasmids can do different things, and they all will have a slightly different purpose in the lab, but broadly that's where we start, the plasmid. And then we have our DNA of interest. So let's say we're talking about the people who wanted to use bacteria to make insulin. So I have this plasmid, and I have the human insulin gene. How do we do that? Put them together? There are a number of different ways, but that is what making recombinant DNA is. We are able to kind of cut that plasmid open and insert our gene of interest. In this case, maybe it's human insulin. We put that plasmid back into bacteria through transformation. We induce transformation. We force this bacterium to pick up DNA from its environment, the plasmid we made. We cause them to become competent even though they don't want to be, and we force them to pick up DNA. How do we do that? Really commonly we electrocute them. It sounds like, okay, so you're putting DNA from multiple organisms together, you're zapping things, like what is wrong with you? And I sound happy about it too. It's just the way it works. Doing a very brief electrical pulse causes little holes to open up in the cell membrane that can allow the plasmid to come in. And then you give them all their, oh, I'm so sorry, right? You give them the right temperature, you give them all their excess nutrients, they get so happy, right? So you can do that, you can do chemical methods. There are ways we can make a cell competent so it'll pick up our plasmid. Okay. So then, what we could do is have all of our clones. Every cell that picked up the plasmid is going to replicate, and in theory, all of the descendants should also have the plasmid. So overnight I go from, maybe 1 or 2 cells that picked up the plasmid to billions that might have the plasmid. That's great. So now I could have a lot of copies of that gene in my clones. And I'm cloning. I promise I'm not cloning. There's nothing to do with people. I'm not cloning people. Or maybe you're just not interested in the gene. You're interested in the protein product. And so again, what you can do is there are plasmids that allow the expression of a protein. Not every plasmid is going to express a unique protein product in this way, but many will. So you induce the expression of the protein, and then you can purify that protein. So this is how you could get your insulin or your human growth hormone. Okay. In this example, maybe I cloned that gene that produces the toxin. So now I have a lot of DNA I have a lot of bacteria, and they all have that same gene that will produce this really cool toxin that will, again, makes me sound terrible, kill all these insects, yay. It's a gene that'll kill all of these insects if I can get it into the plant. So I make a lot of copies, and eventually I try to get that gene into the plant, so then the plant will make the protein for me. This is a true GMO. When you put the DNA from another species into something like a plant or an animal, that's a GMO. So that's kind of the goal here. But again, all roads lead to microbiology. You can't do biotechnology, you can't do this type of work without working with bacteria, without working with microorganisms. You can work with yeast, I guess, that's fine. Microorganisms. So again, hopefully maybe those of you who are like, I'm going to get a degree in biology and I don't know what I want to do, or I'm thinking healthcare, but I'm open to other things. Maybe you see opportunities here where you could look for internships or research positions after you graduate, because you can speak to some of these techniques and how powerful they are. So these are some of the really, really common tools in biotechnology. I wouldn't say that every molecular biologist works with all of these tools. I have worked with all of them, except I've never worked with CRISPR. Just never had the need. I don't modify eukaryotes. I can modify my prokaryotes really easy with all these other tools. But CRISPR is a really powerful tool that I know like Dr. Kong's lab has used to modify yeast. So great tools that any molecular biologist would keep in their toolkit. What is a restriction enzyme? Again, all roads lead to microbiology. I love it because I'm a microbiologist, so I like to inflate the importance of my field as much as possible. So, most people, when you talk about immune systems and immunology, you think of, phagocytic cells that'll come in and, eat the invader. Or you think of antibodies, or the T cells, like the killer T cells that'll come in and destroy everything. Well, bacteria have an immune system too. People don't know that, or they don't think about it. And one part of their immune system are enzymes that are called restriction enzymes. So a restriction enzyme is a nuclease. It's an enzyme that cuts nucleic acids, specifically DNA. And so you might be like, well, what in the world does a bacteria need an immune system for? I just told you in chapter 8, bacteria get viral infections. And when a bacterium gets a viral infection, okay, and even a simple bacterium doesn't want to die. So bacteria have mechanisms to, in some ways, fight off a viral infection. And one of those mechanisms is restriction enzymes. So restriction enzymes are, because I found them in bacteria and we know how cool they are now really critical tools in biotechnology. But again, they are critical tools for the bacterial immune system. They were discovered because they destroy bacteriophage and DNA. And they do that by recognizing a very specific sequence on the DNA. It's sometimes between 4 and 8 nucleotides, a very small sequence of DNA. And when they see that very small sequence, they cut. They're like little scissors. So they cut. So they cut up that phage DNA before the phage DNA can take over the host cell. Does it always work? No. Does your immune system always work? No. Right? Sometimes you get sick. Sometimes the bacteria is going to die, even if it has restriction enzymes. Okay, but sometimes the restriction enzymes allow them to escape. So I told you it's like 4 to 8 nucleotides. The 4 nucleotides, it's very, very likely that the bacterial cell also has those same 4 nucleotides. So how do those restriction enzymes not cut up the host DNA? Well, host DNA is usually methylated. Phage DNA is not. So the restriction enzyme not only recognizes 4 to 8 nucleotides, it also recognizes methylated versus not. So if it sees those 4 to 8 nucleotides and it sees that they're not methylated, it will chop them up and recognize that that's an infection, right? It's DNA that is not ours, so we are going to destroy it. My friend who's an immunologist always gets mad at me when I do this, but I'm gonna do it anyway. It's analogous to like our innate immune system. It just broadly fights the infection that comes in. What if it sees that piece of DNA, it's gonna destroy it. If any of you have taken immunology, right? We have cells in our immune system that just are like, oh, that's LPS, that's bad. I don't care what it is, it's just LPS, it's bad. It recognizes the one molecule that's bad, doesn't matter what cell it's from, it just knows that it's bad. So it doesn't matter what virus it's from, it's like, I know this is bad, I'm gonna fight it. It's like our innate immune system. So it's their innate immune system. These restriction enzymes, again, are very specific. They'll recognize 4, 6, or 8 nucleotide sequences, but the sequences themselves have an interesting feature. All sites recognized by a restriction enzyme are what are called palindromic. So a palindrome is something that can be read the same forwards and backwards, like race car or taco cat. Taco cat is my favorite, but race car works too. If you're like, what are you talking about? Write out race car and then write it out backwards, it's the same. Race car, taco cat, taco cat. Same thing with restriction enzyme recognition sites. So if you look at the top, 5' to 3', G, G, A, T, C, C. The bottom, 5' to 3', G, G, A, T, C, C. It's palindromic. Each restriction enzyme will cut in a slightly different way. Sometimes it will cut and leave what are called sticky ends. These are most useful in molecular biology, if the ends are sticky. So what I mean by that is, let's say that we're looking at BAMH1, and it recognizes G, G, A, T, C, C, and then I have my other side. And what the arrows are indicating is where it cuts. So it cuts right here, and it cuts right here. When it does that, the DNA will come apart. And so the reason they call it sticky ends is because on this strand, right, now that it's cut apart, you have G and then everything that came before it. And then you have this overhang here of G, A, T, C, C, right, and then everything that comes after it. So you've got this little like overhang, and then down here you have the same type of overhang. So it cuts not, it doesn't cut both strands in the same place. It cuts one strand over here and one strand over here. And that becomes really important because if you cut 2 pieces of DNA with the same enzyme, they make the same sticky ends. So if I have my insulin gene and I cut it with EcoRI or BAMH1 and I have sticky ends, I can put it in my plasmid that I also cut with BAMH1 because it has the same sticky ends. But sometimes you have blunt cutters and they just will cut right, they just, cut the whole thing. They don't make a fancy little sticky end. You can use those in molecular biology too. They're harder to work with. So whenever possible, we try to work with sticky ends. Okay. And they're always named after the organism they came from. I would never expect, please don't think I'm gonna be like, what's the sequence for BAMH1 and where did it come from? I would never do that to you. But I mean, even I've worked with these for 20 years and I couldn't tell you what the sequence for EcoRI is. I tell you it comes from E. coli, but that's all you get from me. Okay. So again, right, it leaves these kind of overhangs that are called the sticky ends. Right, so we'll pick up with biotechnology on Thursday. Have a great rest of your Tuesday and Wednesday.